So today we are discussing about the epithelium. The epithelium, they cover the inner surface of the various parts of the body. They cover the inner surface of the various parts of the body. The epitheliums are squamous epithelium, cuboidal epithelium. Types of epithelium are squamous epithelium, cuboidal epithelium, columnar epithelium, pseudostratified ciliated columnar epithelium, pseudostratified ciliated columnar epithelium, ciliated columnar epithelium. They cover the various parts and tissues of the body. They cover the various parts of the body. Epithelia covers the various parts of the body. Okay. So the inner surface of the various parts of the body. And also pseudostratified epithelium, and further we have stratified squamous epithelium, stratified squamous epithelium. Among the stratified squamous epithelium, it may be non-keratinized and keratinized. Non-keratinized and keratinized epithelium. Okay, so these are and last epithelium is the transitional epithelium. Then the last one is the transitional epithelium. Transitional epithelium. Okay. So today we will discuss about these different epithelia. These are varieties of epithelia. Coming to the squamous epithelium, the squamous epithelium consists of a basement membrane. The epithelial cells of the squamous epithelium, they are flattened cells. The epithelium of the squamous epithelium, they are flattened cells, and the nucleus is round and situated at the center. Again, the squamous epithelial cells are nothing but the flattened cells. Okay, the squamous epithelial cells are nothing but the flattened cells. They lie on the basement membrane. This is the epithelial cells, and they lie on the basement membrane or basal lamina, which is usually made up of glycoprotein. All the basal laminas, they are usually made up of glycoproteins. So squamous epithelium is nothing but the flattened cells. Example where the squamous epithelium we can see is, examples are the peritoneum, okay, and also the blood vessels are lined by the blood vessels, the innermost layer of the blood vessels, the blood vessels, part of blood vessels. The blood vessels, they are also lined by squamous epithelium. Epithelium we call it as endothelium. The epithelium we call it as, when surrounding the squamous epithelium which surrounds the blood vessels, we call it as endothelium, and the squamous epithelium which surrounds the squamous epithelium, which is called which surrounds the peritoneum, we call it as mesothelium. Okay, so the squamous epithelium consists of flattened cells which lie on a basal membrane. We can call it as squamous epithelium, and in case of blood vessels we can call it as endothelium, and it is also peritoneum. Peritoneum, it is also nothing but it consists of squamous cells. In that case we call it as serosa. In that case we call them as serosa. And in the esophagus also, the epithelium here is the stratified squamous non-keratinized epithelium. So the epithelium covering the blood vessels, we call it as endothelium. Okay, so squamous epithelium, it is a flattened cells, and the nucleus is rounded and is situated on the basal lamina, made up of glycoprotein. And next is the cuboidal epithelium. Cuboidal epithelium consists of the length and width of the cells is same. Okay, the length and width of the cell is same, and the nucleus is rounded and situated at the center. The nucleus is rounded and situated at the center. We call it as squamous. We call it as cuboidal epithelium. We call it as cuboidal epithelium. Examples of the cuboidal epithelium is, example of the cuboidal epithelium, where example is ovary. We can see in the ovary that epithelium we call it as germinal epithelium. That epithelium which covers the ovary, we call it as germinal epithelium. So the length and width of the cells are same. That is called cuboidal epithelium. And here the cells here are, the nucleus here is rounded and it is situated at the center. Okay. And coming to the columnar epithelium. Columnar epithelium, the length of the cell is more when compared to the width. The length is more when compared to the width of the cell. Here, these are the columnar cells. The nucleus, it is elongated and is situated at the base. Nucleus, it is elongated and is situated at the base, and this columnar epithelium we can see in the stomach. Example of the columnar epithelium is stomach. And sometimes this columnar epithelium consists of cilia. This columnar epithelium, sometimes this columnar epithelium consists of, these columnar cells consists of cilia. And this ciliated columnar epithelium, we can see in the respiratory passages. Example, respiratory passages. Example are respiratory passages. And sometimes what happens is the upper part of the columnar cells show some invaginations. We call them as, the upper part of the columnar cells show some invagination, show some microvilli. These were called microvilli, which is the characteristic feature of the intestine. So this one we call it as brush border columnar epithelium. The upper part of the cell shows some invaginations. We call them as, show some projections. We call them as brush border columnar epithelium. This is the brush border columnar epithelium. Brush border columnar epithelium, which is the characteristic feature of the intestine. In the intestine, we will be able to see brush border columnar cells. Okay, and coming to the pseudostratified ciliated columnar epithelium. Here, the cells are arranged in a single layer, but the cells are of different sizes and shapes. That's why the nucleus placed at different levels. Pseudostratified ciliated columnar epithelium means the cells are arranged at different shapes, and the nucleus is arranged at different levels. The cells are of different shapes. See here. And one cell is like this again, and one cell is like this and again, and one cell. This is the pseudostratified ciliated columnar epithelium. This way the nucleus, it is placed at different levels. Okay, this we call it as pseudostratified ciliated columnar epithelium, and this the cilia is present on the top of the cells, which is best example for pseudostratified ciliated columnar epithelium is trachea. Best example for pseudostratified columnar epithelium is trachea. And next one is the stratified squamous epithelium. Stratified squamous epithelium, it is non-keratinized as well as keratinized. Keratinized epithelium is seen in the esophagus, in the skin, and non-keratinized epithelium is seen in the esophagus. So, what is the structure of stratified squamous non-keratinized epithelium? So, in the stratified squamous non-keratinized epithelium, the basal cells are columnar cells, and outer 3 to 4 layers of polyhedral cells or cuboidal cells. And the outermost layer is the flattened cells. Okay. And the outermost layer is the flattened cells. So, what is the structure of stratified squamous keratinized epithelium or non-keratinized epithelium is the basal cells are columnar cells, outer 3 to 4 layers of polyhedral cells or cuboidal cells, and the outermost layer consists of flattened cells and the nucleus is prominent. So this is the stratified squamous non-keratinized epithelium. Example is esophagus. Okay, example is esophagus. And one more thing is stratified squamous keratinized epithelium. Here in the stratified squamous keratinized epithelium, here we can see the basal cells are columnar cells like stratified squamous non-keratinized epithelium, and outer 3 to 4 layers of polyhedral cells, and outermost layer consists of dead cells. Outermost layer consists of dead cells, and we can't able to see any nuclei. We can't able to see any nuclei in the stratified squamous keratinized epithelium. And the outermost part of the layer consists of substance called keratin. Outermost part of the layer consists of substance called keratin, and we can't able to see any nuclei in the upper part of the cell. Like in the stratified squamous non-keratinized epithelium, we will able to see nucleus here, but in the stratified squamous keratinized epithelium, because the cells as they are growing upwards, what happens here, as they are growing upwards, then cells they will die, and they are replaced by a protein called keratin. Okay, they are replaced by a protein called keratin. Example of stratified squamous keratinized epithelium is skin. So the outermost layers of cells are died and the nuclei can't be seen, and there we can see a substance called keratin in the uppermost layers of the epithelium. So that's the difference between stratified squamous epithelium and stratified squamous keratinized epithelium and stratified squamous non-keratinized epithelium. Okay, that's about the epithelium. And transitional epithelium. Transitional epithelium, otherwise called as urothelium. Transitional epithelium, it is otherwise called as urothelium. It is otherwise called as urothelium. It lines the urinary bladder, ureter, pelvis, renal calyces, pelvis. It lines the urinary tract. That's why we call it as urothelium. Here the cells similar to that of stratified squamous epithelium. The cells are columnar cells, and outer 3 to 4 layers of polyhedral cells are cuboidal cells. Outer 3 to 4 layers of polyhedral cells are cuboidal cells, and the upper cells are umbrella shaped cells, and the outermost layer cells are large cells. They are called umbrella shaped cells. That is the difference between the stratified squamous epithelium from the urothelium. The outermost layer of cells consists of umbrella shaped cells. This is the characteristic feature of the urinary tract. That's why we call it as urothelium. What happens is, when the urinary bladder is stretched, for example, when the urinary bladder is stretched, these umbrella shaped cells, they become flattened. Okay, so umbrella shaped cells are present only in the transitional epithelium. So the innermost layer cells are the basal cells are the columnar cell, and outer 3 to 4 layers the polyhedral cell, and the outermost layer consists of pear-shaped cells, umbrella-shaped cells. These are the umbrella-shaped cells. Okay, this is the umbrella-shaped cells. This is a characteristic feature of the urothelium or transitional epithelium, which lines the urinary bladder, ureter, and also they are also lines the pelvis. Okay, pelvis of the ureter. So that's about the different types of epithelium. So squamous epithelium, it is nothing but a layer of flattened cells, and nucleus is round and situated on the basal lamina. All the epithelial cells, they are lay on the basal lamina made up of glycoprotein. And cuboidal cells, the length and width of the cell is same. Example is ovary. In that cuboidal epithelium, we call it as germinal epithelium. And next is the columnar epithelium. The length of the cell is more when compared to the width of the cell. The nucleus is oval and elongated and situated at the base. Example, stomach. And ciliated columnar epithelium is seen in the respiratory passages. And pseudostratified ciliated columnar epithelium means here the cells are arranged at different levels. That's why the nucleus is placed at different levels, and the nucleus and Along with that, we will be able to see some cilia are present. That's why pseudostratified ciliated columnar epithelium is seen in trachea. Stratified squamous epithelium is of two types, keratinized and non-keratinized. In the non-keratinized epithelium, we have basal cells or columnar cells, and outer 3 to 4 layers are polyhedral cells, and the outermost layer consists of squamous cells. And coming to the stratified squamous keratinized epithelium, similar to that stratified squamous non-keratinized, the basal cells or columnar cells, outer 3 to 4 layers of polyhedral cell, instead of flattened cells, they consist of dead cells, and the dead cells they are occupied by a keratin. Okay. So we can't see any living cells in the upper part of the stratified squamous keratinized epithelium. Best example is skin. And coming to the transitional epithelium, characteristic epithelium of the urinary tract. This way we call it as urothelium. It consists of basal cells called columnar cells, outer 3 to 4 layers of polyhedral cells, and the outermost layer consists of umbrella-shaped cells are present. These umbrella-shaped cells becomes flattened when the bladder is stretched. So this is the best example is the urinary bladder. So these are the different types of epithelia. Which will come across when you are studying any organ, any structure of the body. Okay, so that's about the epithelia. And so today we are discussing about histology of ovary and testis. These are the male and female gonads. They are the male and female gonads of the male and female reproductive organs of the body. Reproductive gonad, nothing but the gonads of the body. Testis is the male gonad and male gonad, and ovary is the female gonad of the body. Okay. So coming to the ovary. So the ovary, it is lined by cuboidal epithelium. The lined ovary, it is lined by cuboidal epithelium, and it consists of cortex and medulla. The ovary consists of cortex and medulla. The ovary consists of cortex and medulla, and the ovary, it is lined by cuboidal epithelium. The cuboidal epithelium which lines the ovary, we call it as germinal epithelium. Tunica beneath the cuboidal epithelium or germinal epithelium, we will able to see tunica albuginea, but it is very thin. But tunica albuginea in testis, it is very thick, and beneath the cuboidal epithelium we will see the cortex. Okay, so in the cortex what we can see is different developmental stages of ovarian follicles are seen. We know that the oocyte, it is surrounded by, we know that the oocyte which is surrounded by flattened follicular cells, we call it as primary oocyte. The oocyte which is surrounded by flattened follicular cells, we call it as primary oocyte. What are oocytes? Because what are oocytes means, usually the oogonia are the cells which are developed from the proliferation of the endodermal cells of the yolk sac. The oogonia are developed from the proliferation of the endodermal cells of the yolk sac. They migrate to the gonads at the 4th week of intrauterine length. These oogonia, they are enlarged and they form primary oocyte. So the oogonia, they become enlarged and they form primary oocyte. Okay, they just enlarge and they form primary oocyte. So when we take the structure of primary oocyte, it consists of oocyte surrounded by follicular cells derived from the stromal cells of the ovary. This follicle, we call it as surrounded by flattened follicular cells. This follicle we call it as primary ovarian follicle. This follicle we call it as primary. The oocyte which is surrounded by flattened follicular cells, we call it as primary ovarian follicles. So we will able to see primary ovarian follicles in the cortex of the ovary. More and more number of primary ovarian follicles can be seen in the cortex of the ovary. Okay, these primary oocyte was surrounded by flattened follicular cells. We call it as primary ovarian follicle. Primary ovarian follicle. So these follicular cells, they are derived from the stromal cells of the ovary. Okay, and this primary ovarian follicle consists of primary oocyte surrounded by flattened follicular cells. Then what happens is the follicular cells, they proliferate. The follicular cells which surrounds the primary oocyte, they proliferate. Okay. The follicular cells which surrounds the primary oocyte, they proliferate. They proliferate. This one we call it as secondary ovarian follicle. The follicular cells which proliferate and they form, this one we call it as secondary ovarian follicle. This one we call it as secondary ovarian follicle. So in the cortex of the ovary, we will able to see secondary ovarian follicles are present. Secondary ovarian follicle nothing but the oocyte surrounded by more layer of follicular cells. The oocyte, this is the oocyte, right, which is surrounded by many layers of follicular cells. The oocyte which is surrounded by many layers of follicular cells, we can see this is the secondary ovarian follicle. So this is the secondary ovarian follicle, the secondary ovarian follicle, okay, which is surrounded by more, the oocyte is surrounded by more and more number of follicular cells, secondary ovarian follicle. Okay, here what happens is the primary oocyte which undergoes meiosis, the primary oocyte which undergoes meiosis and gives rise to secondary oocyte. The primary oocyte which undergoes meiosis and gives rise to secondary oocyte consists of haploid number of chromosomes, Secondary oocyte, the primary oocytes which give by undergo meiosis, it gives rise to secondary oocyte and first polar body. The part of the secondary oocyte receives more amount of cytoplasm and the polar body receives scanty amount of cytoplasm, and it is further, and what happens here is it further degenerates. And one more important thing is between the follicular cells and the oocyte a membrane develops. This membrane we call it as zona pellucida. So zona pellucida appears in between the secondary oocyte and the follicular cells, which appears between the secondary oocyte and the follicular cells. Then on what happens here is, so this is the secondary oocyte, okay, and surrounding the secondary we have zona pellucida, and then what happens here is the follicular cells, they proliferate and proliferate. Then what happens here is some cavities appear in between the follicular cells. Some cavities appear in between the follicular cells, and these cavities coalesce to form one large cavity. So the cavity appears in between the follicular cells. Okay, follicle appears. So a follicular cavity appears in between the follicular cells. This cavity we call it as follicular cavity. And the follicular cells which surrounds the oocyte. So this is a secondary oocyte, right? The follicular cells which surrounds the oocyte, we call them as cumulus ovaricus, and the follicular cells which are present outside, we call this as granulosa cells, and the follicular cells which connect the oocyte to the wall of the follicle, we call these cells as discus proligerus cells. discus proligerous cells. Okay, so what happens is the follicular cells, they exist some spaces, they exist some cavities in the follicular cells, and later on what happens is these cavities coalesce to form one large cavity called follicular cavity, and this follicular cavity is filled with fluid called liquor folliculi. Liquor folliculi. Okay, then what happens here is the follicular cells which immediately surrounds the secondary oocyte, we call it as cumulus ovaricus cells, and the follicular cells which are present outside, we call it as granulosa cells, and the secondary oocyte which is connected to the wall of the follicular cavity by group of cells called discus proligerous cells. Okay, and this is the follicular cavity, it is filled by a fluid called liquor folliculi, and what happens here is the outside the stromal cells, they form one covering outside to the granular cell. We call this covering as the theca interna. Theca interna. Outside to the theca interna, the fibrous tissue appears outside to the theca interna. We call it as theca externa. We call it as theca externa. Okay. So this follicle, we call it as Graafian follicle. So the Graafian follicle consists of secondary oocyte and surrounded by layer of cells as cumulus ovaricus. Outside layer of cells we call it as granulosa cells, and outside to the granulosa cells, stratum granulosum, we can otherwise call it as stratum granulosum cells, and outside to the granulosa cells, the stromal cells of the ovary, they form an outer covering called the theca interna. Outside to the theca interna, a fibrous tissue is present, we call it as theca externa. Okay. So membrane granulosa cells are present here, cumulus ovaricus cells are present here. The oocyte is connected to the wall of the follicle by group of cells called discus proligerous cells. So in the cortex of the ovary, along with the primary ovarian follicle and the secondary ovarian follicles, we will able to see the Graafian follicles also. So Graafian follicles consist of, this is zona pellucida, and group of cells are present around the secondary oocyte, outside layer, and space exists between the inner cumulus ovaricus cells and outer granulosa cells. So we will able to see different stages of development of follicles are seen in the cortex of the ovary. See here, secondary oocyte surrounded by zona pellucida, which is nothing but the glycoprotein membrane, and here are the follicular cells, and outside is also follicular cells because of development of follicular cavity. The follicular cells divides into inner layer and outer layer. So this is the Graafian follicle, theca interna and theca externa. We can't see it clearly during in the slide examination. So but follicular cavity we can easily examine under the microscope. Okay, this is the follicular cavity. This is the Graafian follicle. Okay, then what happens here is the secondary oocyte. The secondary oocyte, at the time of ovulation, it comes from the Graafian follicle and it is taken by the enters into the uterine tube. It enters into the uterine tube by the infundibulum part of the uterine tube. Okay, so the secondary oocyte then ruptures here. The secondary oocyte ruptures here. The secondary oocyte, the Graafian follicle ruptures, and here the secondary oocyte comes out. The secondary oocyte comes out and the Graafian follicle ruptures here and the secondary oocyte comes out. Later, what happens to the follicular cells? Okay, later on what happens to the follicular cells is the follicular cells, they, so sorry, I have to present it with red color, right? All these are follicular cells. So these are nothing but the follicular cells. Okay, these follicular cells usually, but we can't see under the microscope, these follicular cells, what happens is they enlarge and they become polyhedral cells. They enlarge and become polyhedral cells along with some cells from the theca interna, and they form polyhedral cells. Now these cells, these follicular cells, they become enlarged and they become polyhedral cells, and they are filled with a pigment, yellow pigment called, these follicular cells, they are filled with a yellow pigment called lutein. That's why we call them as luteal cells, and the structure we call it as corpus luteum. After the rupture of the Graafian follicle and release of secondary oocyte into the uterine tube, the follicular cells, they become enlarged and become polyhedral. And what happens here is, these follicular cells, they are filled by an yellow pigment called lutein. That's why we call it as corpus luteum. So this is the corpus luteum, which is filled with yellow pigment. So in the cortex of the ovary, we will able to see different stages of development of ovarian follicles are seen. So these are the primary ovarian follicle, which is the oocyte surrounded by flattened layer of follicular cells, and this is the secondary ovarian follicle, the oocyte which is surrounded by a layer of follicular cells. The primary ovarian follicle undergo meiosis and gives rise to secondary oocyte, so which is surrounded by more number of layers of follicular cells. So this is the primary ovarian follicle, okay, which is surrounded by a layer of follicular cells. Primary ovarian follicle, and these are the secondary ovarian follicles. Secondary ovarian follicle, and these are the Graafian follicles. Graafian follicles, okay, and this is the corpus luteum. So we can see the developmental stages of different follicles as seen in the cortex of the ovary, okay. And next is the medulla. Medulla consists of some connective tissue fibers as well as some blood vessels and lymphatics. So this is the medulla, this outer part is the cortex, and this is the medulla. The medulla consists of some blood vessels and also some lymphatics. Some lymphatics are also seen, as well as some blood vessels are also seen in the medulla. So that's about the ovary of the female gonads, and here in the ovary divides into cortex and medulla. The epithelium of the ovary is called germinal epithelium, within nothing but the cuboidal epithelium. On the cortex, we will able to see different stages of development of ovarian follicles are seen, and medulla consists of connective tissue fibers as well as some lymphatics and blood vessels are seen in the medulla, okay. So that's about the histology of ovary, okay. Then we are now going to discuss about histology of testis. Histology of testis. So when we take the section of testes under the microscope, we will able to see testis. It is covered by a layer capsule which divides more number of lobes and lobule, and in each lobule we will able to see sections of seminiferous tubules. Sections of seminiferous tubules are seen. Sections of seminiferous tubules. These are the sections of seminiferous tubules. Seminiferous tubules are seen. So, if you take one section of seminiferous tubule, what we are able to see in one section of seminiferous tubule? So, in the seminiferous tubules, we can see the development of different stages of spermatozoa are seen. Different stages of spermatozoa are seen. That means we can able to see spermatids, and we are able to see secondary spermatocytes, and we will able to see primary spermatocytes are seen. Okay, and spermatozoa are seen. So, various stages of development of sperm cells are seen. Various stages of development of sperm cells are seen in the seminiferous tubule. So, first, what are the structures? Along with the different stages of spermatogonia and spermatocytosis, we will able to see some other cells. We call these cells as the ceratolase cells or sustentacular cells. We can see these cells are irregular shaped columnar cells. We can call it as ceratolase cells or sustentacular cells. These ceratolase cells or sustentacular cells, they provide nutrition to the spermatozoa, and they also helps in the movement of sperms because the secretive fluid which helps in the movement of sperms in the seminiferous tubules, we call which is rich in testosterone. So these sustentacular cells or ceratolase cells, they provide nutrition to the developing spermatogonia. Different stages of development of spermatocytosis, spermiogenesis is different stages of development of spermatogenesis and spermatocytosis, spermiogenesis. We can see here in the seminiferous tubules. First, we know that the germ cells, we call it as spermatogonia. These are nothing but the flattened cells. Okay. Okay. Then these spermatogonia, they give rise to primary spermatocytes, which are large. Each primary spermatocyte, it undergoes mitosis, it undergoes meiosis. Each primary spermatocyte, it undergoes meiosis to give rise to secondary spermatocyte, which are small comparative to the primary spermatocyte. Each secondary spermatocytes undergo meiosis 2 and gives rise to spermatids. These spermatids, they undergo transformation and form the spermatozoa. So the basal cells are the spermatogonia cells, and outer we have primary spermatocytes. Then you have to draw secondary spermatocytes, which are comparatively smaller, and then spermatids, which transform into spermatozoa by process of spermiogenesis. Different stages of development of spermatogonia are seen in the sections of seminiferous tubules. For example, there are the ceratolase cells, and we have to draw spermatogonia here. Out of that, we have to draw spermatocytes and secondary spermatocytes, spermatids, and we will have to draw spermatozoa. So these are the flattened cells, primary spermatocytes, secondary spermatocytes, spermatids, and spermatozoa. So we have to draw the different stages of development in each section of seminiferous tubule. So that's the section of seminiferous tubules. So in the section of testis, we will be able to see section of seminiferous tubules. In the seminiferous tubule, different stages of development of spermatocytosis, spermiogenesis or spermatogenesis is seen. Okay. That is nothing but spermatogenesis. Different stages of spermatogenesis are seen, and here we have the basal cells are spermatogonial cells. They undergo mitosis, which gives rise to primary oocytes, which are larger cells, and these primary oocytes, they undergo meiosis and form secondary spermatocytes. These secondary spermatocytes, they undergo second meiotic division and gives rise to spermatids. These spermatids, they are transformed into spermatozoa by a process called spermiogenesis. Along with the germs, developing germ cells, we will be able to see some irregular shaped columnar cells or cells are seen, that is called, and that one we call it as sustentacular cells or ceratolase cells. These sustentacular, when you take the one sustentacular cell, in the presence of sockets are present, and within the sockets we can see the germ cells are seen here within the sockets, and the sustentacular cells, they provide nutrition to the developing germ cells, as well as they secrete fluid which is rich in testosterone. So that's about the histology of testis. Okay.